Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Before we jump in, please take a moment to like this video and subscribe to our channel. It really helps support the Sand Science Experience and allows us to keep bringing you these deep dives into groundbreaking scientific discoveries. Today, we're embarking on a deep dive into a truly groundbreaking study. This research offers some profound new hope for a really devastating neurological condition by focusing on a vital molecule in our bodies. Coenzyme Q10 or CoQ10.
Yeah, it's fascinating stuff because for years, despite CoQ10's absolutely critical role in making energy and acting as an antioxidant, trying to boost its levels, especially where it counts, like in the brain, it just hasn't worked very well. This new research really tackles that fundamental challenge head on with a genuinely novel approach.
Exactly. So we're going to unpack why CoQ10 is so indispensable, explore why the traditional strategies have fallen short, and then really dig into how this new scientific understanding could fundamentally reshape how we approach certain mitochondrial diseases.
It's about moving beyond just simple supplementation, right?
Precisely.
Okay, let's get into it. CoQ10, it's kind of an unsung hero inside our cells, wouldn't you say?
Definitely, especially in the mitochondria.
The powerhouses, yeah.
Right, they generate most of the cell's energy, ATP. And CoQ10 is essential for that process. It powers pretty much everything. Basic cell function, complex thoughts, everything.
Its importance is often underestimated, I think.
For sure. I mean, we know low CoQ10 causes rare but severe primary deficiencies, leading to things like encephalopathy, basically serious brain dysfunction.
Right.
But its decline is also linked, maybe more subtly, with common neurodegenerative disease, like Parkinson's, Alzheimer's, even just the natural aging process itself. Its influence is really quite pervasive. Okay, so if it's that critical and low levels are bad news, why haven't we just been taking supplements? I mean, you see CoQ10 everywhere in health stores.
That's the million-dollar question, isn't it? And it points to the core problem. The fundamental issue is that CoQ10, when you swallow it, has really poor bioavailability.
Meaning it doesn't get absorbed well.
Exactly. Not enough gets into the bloodstream, and even less makes it into critical tissues, especially the brain, because of the blood-brain barrier. So even though we've wanted to boost CoQ10 levels for ages, it's been stubbornly resistant to just taking more pills. We couldn't get enough where it needed to go.
Right. Okay, so the delivery was the bottleneck.
Pretty much.
This is where the story gets really interesting then. The paper we're looking at highlights a recent, absolutely pivotal discovery that kind of flips the script. Instead of trying to force the finished CoQ10 molecule into cells, they're looking at how our bodies make it naturally, specifically focusing on its head group.
Precisely. It's a strategic shift. Think of it like instead of trying to deliver the finished car, you focus on fixing the production line. The key enzyme here is called Hpdl. It helps make something called 4-hydroxymandylate, or 4-HMA.
Okay.
And this 4-HMA then makes 4-hydroxybenzoate, or 4-HB. Both of these are direct precursors, building blocks for the CoQ10 head group.
So they're like the specific ingredients the body needs to assemble its own CoQ10.
You got it. They're finding the missing pieces for the internal factory.
And what makes these two, 4-HMA and 4-HB, better than just taking CoQ10 itself?
Their size. That's the crucial difference. They're both small molecules, under 200 Daltons. Which means they can be easily taken up by cells, they can cross membranes readily, and then get efficiently slotted into the CoQ10 synthesis pathway inside the cell. It completely bypasses that whole bioavailability problem we talked about. It's like finding a side door.
That makes a lot of sense. So how did they test this idea? Did they just work in cell cultures?
No, they went further. They used a really sophisticated mouse model, one that very accurately mimics a devastating human condition tied to this very pathway.
Okay, tell us about that.
Yeah. They studied mice called Hpdl mice. These mice lack the Hpdl enzyme, and they effectively model an ultra-rare but lethal human mitochondrial disease. It's called neurodevelopmental disorder with progressive spasticity and brain white matter abnormalities, or NEDSWMA for short.
Wow, that sounds severe.
It is. These mice show severe neurodevelopmental problems, spasticity, muscle stiffness, seizures. And tragically, if you don't treat them, they die by about postnatal day 15.
15 days. That's incredibly fast.
It is. And the researchers confirmed these mice had the same metabolic markers as the human patient's low 4-HMA, high lactate to pyruvate ratios. It's a really good model.
So what happened when they treated these mice with the precursors, 4-HMA or 4-HB? The results must have been striking.
Striking is an understatement. They started oral treatment really early, between day 3 and day 7 after birth, and the result. An astonishing 90 to 100% of these treated pups survived to adulthood.
From near certain death by day 15 to a normal mouse lifespan.
Exactly. A normal mouse lifespan, 18 months or more, compared to 0 survival in the untreated group. And crucially, this really drives the point home. They also tried treating some pups with conventional CoQ10 supplements.
Yeah.
No effect whatsoever. They still died by day 15. It really highlights that this new approach, targeting the synthesis pathway, is fundamentally different and effective.
Okay, survival is obviously huge, but did the treatment actually fix things at a cellular level? Did it repair the damage in the brain?
It absolutely did. Giving these intermediates successfully restored the production of CoQ9 and CoQ10 right there in the brains of the treated Hpdl mice. And this led to significant visible improvements in the brain structure, especially in the cerebellum, which is hit hard in these disorders.
What kind of improvement?
They saw things like mitochondria getting bigger and looking healthier within the cells. They saw Purkinje cells, key neurons in the cerebellum, regrowing their complex dendritic branches, those tree-like structures they use to communicate. And importantly, they saw a dramatic drop in cell death. They used Tunnell staining to measure this and saw less cell death across the cerebellum, cortex, striatum, thalamus, key brain regions.
So it wasn't just keeping them alive, it was actually reversing some of the underlying structural damage.
Yes, exactly.
And what about function? Do the brains actually start working better? Structure is one thing, function is another.
Great question. And yes, they did function better. The researchers looked at mitochondrial function directly. Electron flow through complex 1 and complex 2 of the respiratory chain that's core to energy production was severely messed up in the untreated mice.
Okay.
But the 4-HMA treatment restored this electron flow back to near normal levels, even when they checked in adult mice. So fundamental energy production was back online.
That's huge.
It is. And they also looked at the electrical activity of those Purkinje cells in the cerebellum. The treatment corrected abnormal firing patterns and helped the cells mature functionally. Essentially, the cells started behaving normally again, communicating properly. This translated into better motor coordination, fewer seizures in the mice, real functional recovery.
The mouse results are just incredibly compelling. But, the big hurdle is always translating this to people. Does the paper give us any indication there?
It absolutely does, and this is perhaps the most powerful part of the study. They include a case study, a human patient.
Yeah.
They describe an 8-year-old child. This child had rapidly progressing spasticity, and the cause was found to be the exact same genetic variants in the Hpdl gene as the mouse model. And adding to the urgency, his 2 younger siblings had tragically already died in infancy from a similar catastrophic version of the disease.
Oh, that's heartbreaking. So a desperate situation for this family.
Truly desperate. They had tried conventional CoQ10 supplements for about 6 weeks.
And it didn't work.
No effect. Just like in the mice. So they got FDA approval for what's called a single-patient IND investigational new drug application to try the oral 4-HB treatment.
Okay, so they started the 4-HB.
Yeah.
What happened over the next months?
Over about 250 days, roughly 8 months, the improvements were significant. And importantly, they weren't just subjective feelings, they were objectively measured using standard clinical scales.
Can you give us some examples of those measurements?
Sure. They used the modified Ashworth Scale to measure spasticity, that muscle stiffness. His score went from 10 down to 6. That's a clinically meaningful reduction in stiffness.
Okay, that's a real change.
Definitely. And the Spastic Paraplegia Rating Scale, which looks at how spasticity impacts daily life, improved significantly too, from 13 down to 7. Even his hand-finger dexterity, measured with something called the 9-hole peg test, either stabilized or maybe even subtly improved, which is remarkable for a disease that's supposed to be progressive.
Those numbers are impressive. But what about the things that really matter day to day? How did it change his life?
That's where it gets even more compelling. His endurance improved dramatically. He went from barely being able to walk across a hospital lobby to walking for 20 minutes straight.
Wow.
His balance got much better. He couldn't even stand with his feet one in front of the other initially, but within 2 weeks he could. After 8 months, he could step sideways to catch a ball. Falls used to happen multiple times every single day. They became rare.
That's life-changing for him and his family.
Absolutely. And maybe the most powerful thing, within about 6 weeks of starting treatment, he could run again. And eventually, he could ride a bicycle.
That's just incredible.
It really is. And what's fascinating is that these huge functional gains happened without major changes showing up on his brain MRI scans.
What does that suggest?
It suggests the treatment was reversing functional problems at the cellular level, maybe restoring neuron communication and energy production, even if the larger scale structural damage wasn't immediately visibly repaired on the MRI. It points towards a real functional recovery.
Truly remarkable. So, stepping back and looking at this whole deep dive, what are the big takeaways, the broader implications here?
Well, first, it fundamentally proves that the CoQ10 synthesis pathway in mammals can be pharmacologically fixed using these head group intermediates. That's a huge conceptual leap. Second, it really hammers home the idea of a critical window for treatment.
Right. You mentioned that with the mice. Treatment had to start super early.
Exactly. Before postnatal day 10 for them to survive. This strongly suggests that for these kinds of neurodevelopmental conditions, intervening early during brain development is absolutely crucial. Now, for humans, especially if they have some leftover Hpdl enzyme activity, that window might be a bit longer, which offers hope. But the principle remains: early diagnosis and treatment are key.
It was also really interesting that even partially restoring CoQ levels seemed to be enough for this massive rescue effect in the mice. You didn't need 100% restoration.
Yeah, that points towards a threshold effect for CoQ. Maybe you don't need perfect levels, you just need to get above a certain critical point for mitochondria to function well enough and see these big neurological improvements.
And that has implications for treatment, right?
Absolutely. It opens the door for optimizing doses, maybe developing better formulations, and it really strengthens the argument for newborn screening for these kinds of metabolic errors. If you can catch it at birth and start treatment immediately, you maximize the chances of a good outcome within that critical window.
You also touched on some nuances, like the male mice still having some issues even with treatment.
Right, that was interesting. Male mice still had an altered respiratory exchange ratio, suggesting some metabolic differences, and lower grip strength compared to females, even when treated. It suggests that while the treatment is incredibly effective, maybe life-saving, the current way they did it might not be perfectly optimized yet. Or perhaps certain features of the disease are just harder to reverse completely.
Shows there's still more to learn, more refinement possible.
Always. That's the nature of science. But the foundation laid here is huge.
This deep dive really does reveal a truly transformative approach, taking something previously untreatable and offering real hope. It's such a powerful example of how digging into fundamental biology can unlock completely new ways to help people.
I agree. What really stands out to me is the potential for precision medicine here. You identify the exact metabolic block in a patient, and you design a specific small molecule to bypass it. And the results can be profound, real-world benefits. It really makes you wonder, doesn't it? What other diseases, maybe even common ones linked to subtle CoQ10 issues, might now be treatable with similar targeted approaches? That's the exciting next question.
Absolutely. What specific aspect of this breakthrough stands out most to you, thinking about the future? We really hope this deep dive has given you a clearer, more informed perspective on this incredibly exciting area of scientific discovery. Remember to like this video and subscribe to the Science Experience for more deep dives into groundbreaking science. Thanks for joining us.